Hello lovelies, in this video we're going to be going through the AQA A-Level Biology paper 1 that we've written for 2024. Now don't forget, these topics could come up on paper 1 or they could come up on paper 3. So it is well worth doing paper 3 and paper 1 before you go into paper 1. But here is paper 1, so without the big S.
The image shows a pollen grain. State which type of microscope was used to take this image and explain your answer. So hopefully we know from the image it's definitely an electron microscope. So when it says type of microscope, not only is it asking if it's an electron microscope, but what type of electron microscope it is. So this is going above and beyond what you would have done at GCSE. So we also need a reason why we're choosing this type of electron microscope. So hopefully you can tell this is a 3D image and it's of the external surface of the pollen grain. So those two clues are enough to tell us that it should be an SEM, or a scanning electron microscope image. Only TEM is used for cross sections, 2D cross sections of internal structures, whereas SEM is used for 3D external kind of structures. So that could be our reason. So we're going to put it's a scanning electron microscope, or an SEM was used as the image is in 3D. Calculate the length of the pollen grain. Okay, so we don't get given the magnification in this question. Instead, we've been given a scale bar. Sometimes the magnification will be printed on the picture, sometimes you'll get a scale bar, sometimes you'll be told the magnification of the question, but because this is A-level, they've made it slightly harder with the scale bar. So we just have to do two lots of the equation that we would normally use. So magnification equals the image size over the actual size, and then we're going to need to use the actual size equals the image size over the magnification. And we're going to have to use the scale bar to calculate the magnification first. Then we're going to measure our pollen grain with our ruler and use our other equation to calculate the actual length. So there we go. You can see my ruler here. I've measured the scale bar and it is 12 millimeters. So my magnification equals my image size. So that's the length of the scale bar on my piece of paper. That's my image of the scale bar, which is 12 millimeters. And I'm going to divide that by 4 micrometers, which is the actual size that that scale bar represents at the magnification of this image. I can't do that division because I'm going to need to convert first so that my units are the same. We times by 1,000 to convert 12 millimeters into 12,000 micrometers, and that gives us 3,000 times. So the magnification of the image has been magnified 3,000 times from the original size. Now I can do my actual length. So I've not shown you a picture of this, but I've measured the pollen grain at its longest point, and it's about 45 millimeters. Probably they don't really allow much leeway at this because they've measured it and know that it's 45 millimeters. So you normally have to get it spot on. So we're going to do 45 millimeters divided by 3,000, which is our magnification, and that gives us 0.015 millimeters, or in micrometers, which is slightly better, 15 micrometers. So this is a little bit more of an extra calculation than we would normally do for one of these, but what it does is it shows that we can use and rearrange our magnification equation, which we have to know and learn, and we can use our ruler and measure and convert as well. So we've got lots of marks in there. So make sure you show all this working so that you can get marks even if your measurements weren't 100% on point. Also, obviously, your measurements are going to slightly vary from mine, potentially, depending on how you printed this out. So if you did all of this correct working, but your measurements were slightly different, then obviously you can have the marks here. High power microscopes must also have high resolution. Explain what is meant by the term resolution. So explain what is meant by the term. Questions are basically just a fancy way of saying define. So define resolution basically for 1 mark. So hopefully this is a definition that we know. Resolution is the ability to see 2 objects as separate objects or as separate points. You can also have a mark for saying it's the ability to distinguish between 2 objects. Pollen is produced in the male reproductive organs of flowers. A cell from the plant this pollen grain was collected from had 36 chromosomes. State how many chromosomes would be found in the pollen grain. So this is a meiosis question. So The diploid cells, because they will be coming from the body of the plant, are 36 chromosomes, whereas the gamete, which is the pollen, will be haploid. So they've told us that in the diploid cell it's going to be 36 chromosomes. So we just have to half that number to get the haploid cell because there'll be half the number of chromosomes in the gamete. So the answer is 18. The diagram shows the mechanism that amino acids can be absorbed through an epithelial cell from the small intestine into the blood. So the key shows us that the circle is the amino acids. There's one coming in from the lumen of the small intestine into the cell, and then there's one leaving the cell and going into the capillary. Explain 2 adaptations of this cell that allow it to carry out its function efficiently. So it tells you this cell is an epithelial cell from the small intestine or from the ileum. It might describe it as, you may have learned, it's an ileum epithelial cell. So what adaptations has this cell got to help it carry out absorption? So this is its function. It's a cell that absorbs all the molecules, not just amino acids, but that absorbs the other molecules from the lumen of the small intestine and into the blood. So it needs to explain the 2 adaptations. So we're not just stating them. For 2 marks, we need to explain why each adaptation we're going to mention helps it to carry out its function efficiently. So there's 2 kind of obvious ones that we can get from the diagrams. If you didn't know this already, or it wasn't off the top of your head, we've got the microvilli at the top of the image. And the reason why that obviously helps with absorption is because it increases the surface area of the membrane that's in contact with the lumen. And then we've got multiple transport proteins. So these will be carrier proteins, or they will be co-transport proteins. So we've got 2 co-transport proteins, or a sodium-potassium pump, as we can see in the bottom right corner with C. So lots of those, more than maybe a normal cell might have, in order to be able to carry out either active transport or facilitated diffusion, or co-transport. And then also we've got the sort of symbol there, we've got the ATP being hydrolyzed, so that's showing you that that's being used by the sodium-potassium pump. So in order to provide a lot of energy for that, so the ATP that we need for the active transport, you're going to need lots of mitochondria. And this is the thing that people get wrong with this question a lot, is that they just say mitochondria. But that's not an adaptation. Every animal cell will have mitochondria. The adaptation is that you need lots of, or many, mitochondria because you're using more ATP than usual. So any of those, plus there's some others in the mark scheme as you can see, but I would go with microvilli because it's a nice one to increase the surface area for absorption, and then the many mitochondria to produce the ATP needed for active transport. Explain why the ileum contains both endopeptidases and exopeptidases for efficient digestion of proteins. So it's an explain why question, so we need reasons why we need both of these 2 different types of enzymes, and how does that help us digest proteins more efficiently. So endopeptidases, we should know, hydrolyzes internal peptide bonds, whereas the exopeptidases hydrolyzes the terminal peptide bonds. So why does having both of these enzymes make digestion of proteins more efficient, or otherwise sort of faster? And if we think about the fact that the exopeptidases hydrolyze terminal bonds, that means they hydrolyze the final bond at each end of a protein, a polypeptide chain, and that releases then that single amino acid, which can then be absorbed. And so hydrolyzing the internal bonds, so splitting a polypeptide chain into 2, creates more terminal ends for exopeptidases to work. So if you have one single polypeptide chain, it will have 2 terminal ends. That means there's 2 terminal points that the exopeptidases could work to produce the single amino acids. But if you break that polypeptide chain in half, which is what the endopeptidase does, then you've now got 4 terminal ends for the exopeptidases to work on. So that means that 4 terminal amino acids can be hydrolyzed and broken off and then absorbed instead of 2. So the more, both of those working together kind of increases the speed of that digestion of those proteins. So those are the 2 marks. We need to give 1 mark for describing how one of these enzymes works. We don't have to say both to get the mark, just one. So I'm going to say The endopeptidases hydrolyze internal bonds of proteins. And you don't have to say the word peptide bond here either to get the mark. And then the second mark is for saying why is it more efficient, having both of them. So by having the endopeptidases, it creates more terminal ends for the exopeptidase to hydrolyze and release single amino acids. Again, you could have said it increases the surface area that exopeptidases could then work on, and you don't need to specify about the release of the single amino acids to get the second mark. Describe the process occurring at A. So if we look at A, that's showing the co-transport protein. So we've got a sodium ion coming into the cell, and we've also got an amino acid coming into the cell. So hopefully we know that it's the sodium ions that are travelling down their concentration gradient, so diffusing into the cell, and it brings along an amino acid for every sodium ion that comes through the transport protein. So it's just a describe question, so it just needs us to say that. So what is happening here? Describe what's happening. So the sodium diffuses in down the concentration gradient. That is important for the mark because we've got to sort of show that actually the sodium ions are moving in down the concentration gradient and they're bringing the amino acid regardless of the concentration gradient of the amino acids, because this means that we can almost take them into the cell and then into the blood against their concentration gradient if need to. And so we just need to say co-transport has to be in your answer somewhere, obviously, because this is what we're talking about. And so it co-transports an amino acid in with each ion. Scientists discovered this mechanism after treating ileum cells with a respiratory inhibitor. Explain why absorption of amino acids would stop if the cells were treated with a respiratory inhibitor such as cyanide. So when we see this idea of a respiratory inhibitor, we have to know that that's inhibiting or stopping respiration, which means that it's going to stop the production of ATP, or the amount of ATP available in the cell is going to decrease. So for the question to get the marks to answer, we need to explain why no more amino acids will be absorbed into the cell or into the blood if there is no more ATP available. So no ATP available means we can no longer carry out active transport. And the part of the cell that carries out active transport is part C, the sodium-potassium pump. So if that doesn't happen, then no sodium is being pumped out of the cell into the capillary, which means we don't get a lowering of the concentration of the sodium ions inside the cell. And that creates the concentration gradient. So it creates the fact that there's lower concentration of sodium inside the cell than outside of the cell in the lumen of the small intestine. So that means that we haven't got that concentration gradient for sodium ions to diffuse down into the cell anymore. And we know that we need that to bring along and bring in the amino acids into the cell. So this is kind of a full understanding. Do you understand how co-transport of amino acids happens? And it's that typical AQA, A-level style question where they're asking you, if this doesn't happen, what's that going to stop? Or explain how that's going to stop this process. And that's basically what we have to do. So just talking about co-transport and explaining it, but kind of backwards. So this won't happen, so this won't happen, so this won't happen. And that's what we're going to get for the 3 marks. So talking about the lack of ATP, linking that to the lack of active transport, linking that to the lack of concentration gradient, and then that's the reason why we don't get amino acids moving in. So I've written this out kind of full sentences. You could do sort of shorter points, as you can see in the mark scheme, you'd still get marks. There is often marks sometimes for this where if you just described co-transport as it should happen, but make a sentence or make a point that's saying, because there's no ATP, this won't happen, then that can also get you marks as well. So the sodium-potassium pump requires energy from ATP to pump sodium ions out of the cell. No ATP means this stops, so the concentration of sodium ions is not lower inside the cell than outside. I could have just said no concentration gradient created there. Absorption of amino acids requires the facilitated diffusion of sodium ions into the cell, which no longer occurs as there is no concentration gradient. So you can see I try to say there's none of this, and so this won't happen. There's none of this, so this won't happen throughout that. But as I said, if you said normally this would happen and describe actively co-transport with these 3 kind of points, but then said because there's no ATP, this won't happen, then you can kind of get some marks for that, saying that as well. The fluid mosaic model is used to characterise the plasma membranes found in all organisms. Describe the fluid mosaic model. So what is it? What is the fluid mosaic model? Why do we have the words fluid and mosaic? Why do we use them to describe it? So what does that actually mean? So the reason we use the word fluid is because it describes the fact that the phospholipids are able to move around within the bilayer. And the mosaic part comes from this idea that there's proteins studied randomly throughout the membrane. So we've got this idea of a mosaic pattern of proteins within the bilayer. So it's a describe question. We just need those 2 points. So describing what it is. So it's a phospholipid bilayer. The phospholipids are free to move within the bilayer, which is why it's described as fluid. And there's proteins studied or randomly placed throughout the bilayer, and that's where the mosaic term comes from. This is a common question, and being able to describe the fluid mosaic model is something we have to be able to do. So this is a good way of kind of making sure your language and the way you describe this is accurate. So the membrane is described as fluid because the phospholipids are free to move, and the mosaic structure because of the proteins spread throughout the phospholipid bilayer. You'll notice the phospholipid bilayer is underlined in the mark scheme. It doesn't actually matter whether you put it in for the first point or the second point, but as long as you've used that phrase, because that is how the majority of the membrane, the structure of the membrane is formed. Betalans are water-soluble pigments present in vacuoles of plants. The most common is betanin, which can be found in red beetroots. The structure of betanin is shown below. Explain why betanin cannot cross the plasma membrane. Okay, so got the diagram here. We've got some information about it in the top of the question. So we need to use that or just use our knowledge. So this is something that whether or not they've given us a diagram and whether or not they've given us information about this, you can just use the knowledge you've got as to why some molecules can't cross the plasma membrane. So can't move through the plasma membrane. So it tells you it's water soluble. That tells you that it's charged or polar. You can see some charges, positive and negative, on the diagram as well. So that tells you that it's hydrophilic, it's charged, it's polar, it's able to dissolve in water. Therefore, it's not lipid soluble, which means it will be repelled by the hydrophobic sections of the phospholipid bilayer. So it can't just go through. Other than being charged or polar molecule, which is some properties that stop molecules from being able to move through the phospholipid bilayer, the other reason is that molecules can be too large. So if you're too large, you can't just diffuse through. Both of these would prevent it from passing through the phospholipid bilayer. They would need a specific carrier protein or channel protein in order to be able to move through it. So that's what we're going to say for 2 marks. We need to give a reason, one of the reasons, either that it's too large or that it's water soluble or charged or polar, one of those, and then explain why that means it can't cross the membrane. And you could obviously include the idea that it would need to use a channel protein instead of being able to diffuse through. So the betanin molecule is polar, so it cannot pass through the hydrophobic phospholipid bilayer. So I get 2 marks for this because I've talked about the fact that it's polar, and I've mentioned the fact that it would not be able to get through the hydrophobic part of the phospholipid bilayer. Or you could say it would be repelled by the hydrophobic part of the phospholipid bilayer. You've got lots of options within the mark scheme that you can use here to get the 2 marks, as long as you've given kind of why it can't pass through, so your reasoning. So the structural feature, either polar or large, and then why that means it won't be able to pass through. A student carried out an investigation into the effect of ethanol on the permeability of plasma membranes in beetroot cells. They used the following method. They used a cork borer to cut out cylinders of beetroot. They rinsed the cylinders in distilled water. They used the serial dilution method to produce different concentrations of ethanol. They placed the beetroot cylinder in 100 millilitres of each concentration of ethanol in test tubes in a water bath at a controlled temperature for 20 minutes. They then transferred the solution from each test tube into a cuvette and measured the absorbance using a colorimeter. So the independent variable here is the different concentrations of ethanol. So hopefully we recognise this as the kind of membrane permeability practical that we should have done. And this is about making sure that we're changing just one thing. So we're changing the concentration of ethanol, we're using a solvent. And then everything else it says here has been kept the same in terms of the temperature being controlled with the water bath, the time they were in the water bath for, which was 20 minutes, and then the volume of water, or ethanol, sorry, that was in the test tubes has all been kept the same. So the question wants us to state 2 other variables the student should have controlled in this experiment that are not mentioned in the method. So not these ones, not the temperature, not the time that the solution, the chips are in the solution, or the volume. So we need to think of something to do with the beetroot, because that's the only thing, other than saying they cut out the cylinders, they didn't specify anything about the beetroot cylinders. So if you remember from doing this practical yourself, we need to keep them the same length or volume, so use the same cork borer and then cut them all to the same length. So they've got this same shape, size or volume. They could have made sure they rinsed the cylinders in distilled water for the same length of time. We know that obviously washes off excess pigment from around the discs, but obviously you could rinse and rinse and rinse and rinse and rinse for ages. So it depends on how long they rinse that for. Also making sure we're taking cells from the same type of beetroot or the same beetroot itself, so that all those cells and all that tissue should be the same. So we're comparing different chips, but all of the cells and everything should be the same, and the cytoplasm concentration, things like that should be the same. So I'm going to say they should have used cylinders from the same beetroot, and then talk about the length being the same. But you've got lots of options on that mark scheme you can use. The student is provided with a stock solution of 95% ethanol. Using calculations, show how they would produce the 100 millilitres of the 20% ethanol solution needed for the investigation. Okay, so this is just a serial dilution question, or a question about dilution and calculating how much you would need to dilute a solution to get it to be the right concentration. So for any question like this, we're going to use a nice easy method that I always go to, because I find these questions sometimes can be quite hard to get your head around. So we're going to use the C1 times V1 equals C2 times V2 method. So C1 is the start concentration, V1 is the start volume, so that's sort of like the stock solution and how much you make it up with. C2 is the final concentration and V2 is the final volume. So what volume do you want to end up with at the end, and what concentration do you want to end up with at the end? What solution are you making? So we've got the 20% ethanol solution and 100 mils of it. That's our final concentration and final volume. We know the concentration of the start, which is the stock solution, that's 95%. So all we need is we need to figure out what the volume of that stock solution we need to make up our 20% solution to start with, and then we will need to obviously dilute that with water. So we need to rearrange for V1, which means we get C2 times V2 divided by C1. So now we just need to put those numbers into that equation, and then hopefully we'll find V1, and then from that we can find out how much water we would need to add. So when you're working with percentages in this equation, we should use decimals. So V1 equals 0.2 times 100 divided by 0.95. That gets us 21.05 millilitres of stock solution. So that's how much stock solution you would need to take, the volume you need to take, put into a new tube or beaker, and then now you need to obviously make that up to 100 mils with the distilled water. So we would obviously need to just work out how many to add. You'd obviously, if you were a chemist, you could have done this in a volumetric flask, 100 mil volumetric flask, because there's a meniscus line on there where you can make it up to exactly 100 mils. Or for this question, we need 2 marks, so we need to say the volume of distilled water we're going to add to our 21.05 mils of stock ethanol, and then that will give us 100 millilitres of 20% ethanol solution. In the space provided, plot a graph using the data in the table. So we've got the table that shows the results, we've got the ethanol concentration percentage, and then we've got absorbance in arbitrary units as well, and that's the mean absorbance from those repeats. Okay, so axes first. So we're going to have ethanol concentration on the x-axis, and that's going to be our percentages. When you're doing these graphs in the exam, most of the time the axis should be very simple and the space they've given you to draw the graph will fit the graph that you need to draw and will fit the axes on if you're using a sensible linear scale. So there shouldn't be not enough space or not enough space to draw the graph, and it should mostly fill the space you've been given. So you shouldn't end up with a tiny graph in a corner, and you shouldn't have to end up needing to like extend your axes or anything like that. If you're doing that, you've probably picked a weird scale or you're doing something wrong. So absorbance is going up the y-axis, and it's just arbitrary units. So for this one, I've got to go from 0.02 to 0.6, nearly 0.7. So luckily, if you go up this in 0.1s, obviously each one of these tiny squares will then be 0.02. So that is perfect for what we need it for. And we can get all the way up to 0.7, which is just above where we need it to go for. I hadn't actually put in my axis lines, so I've done that now, and obviously you should do that with a ruler. So now we've just got to plot our points. Obviously we need to do this accurately, so that first one, as we said, is 0.02, because one of those squares is worth 0.02, because we're dividing 0.1 by 5. So that tells us what each of the tiny squares is worth, so that goes with the 20 concentration. Then obviously 0.3, we're just going to go in between the next two squares. You only get tolerance of half a square, so that is the worth doing, making sure you're really accurate with plotting these points, otherwise you may lose marks. So then we have 0.06, so we just need to go up 3 tiny squares. Then we're going all the way up to 0.66, so we need to go 3 tiny squares up from 0.6, and then we've got, again, just half a square up for the 100% concentration. And then we just do our line of best fit. I've done a smooth curve through these points. That is how best to do this data. And then I'm just going to go back and double check I've done all of the things that I need to get marks. I've got my axis titles. I've included the units in brackets. Make sure you don't forget that. Just copy exactly what is in the table title for your x-axis and y-axis. Do exactly as it is in there with the units. I've got my linear scales, which work, and they're fine. I've not accidentally skipped a number or something. I've plotted my points. I'm happy that they're accurate. I've done crosses, which is very important, so that the examiner can see exactly where the middle of my cross point is, exactly where I've drawn those marks, even when my line is drawn through them. If you do dots with a pen or a pencil, one, you can't tell clearly where the middle of the dot is, and you may not get it accurate, especially if you're trying to make them bigger. And then when you draw a line of best fit through them, those dots can completely disappear and be really hard to see. So try and make sure you use crosses at all times, and then your line of best fit should go through the middle-ish of those crosses. It's not going to be 100% perfect, and mine's obviously a little bit wobbly, but it's plotting the pattern that the data is showing, and that's what's most important about your line of best fit. Explain the pattern shown by the graph. So we need reasons to explain why this pattern has happened, and the pattern that we've got is that as we increase the absorption, so as we've increased the ethanol concentration, the absorption has increased. What does that actually mean? Well, it means more pigment has leached out of our cells. Why has more pigment leached out of our cells? Well, it's because the membrane has been made more permeable by that ethanol concentration. So we've got to link the increase in ethanol concentration to the increase in absorbance, and that is linked by the fact that there's more pigment leaching out because the ethanol, the increase in concentration of ethanol, has made the membrane more permeable. And that's what it wants to see here. It wants to see that link, that you understand what the ethanol has done, and that you understand that idea of being more permeable of a membrane means more pigment will leach out, which gives that higher reading on the colorimeter because there's more pigment in that solution for the light to try and pass through. So as the concentration of ethanol increased, it disrupts the phospholipid bilayer, making the membrane more permeable. This causes more pigment to leak out of the cells, which increases the absorbance. So I've written mine maybe put it back to front from the mark scheme. So I think that makes more sense to me to say as the ethanol concentration increases, then the absorbance increases. That would be the description of the pattern. So I've used that as to frame my explanation. So as the concentration of ethanol increases, what does it do? And why does this cause the increase in absorbance? Because more pigment leaches out of the cells. So hopefully that makes sense, and that shows you how it's kind of the easy way of thinking about how to get those 2 marks. Hand sanitizer gels work because they contain ethanol. Effective hand sanitizers should contain a minimum of 60% ethanol, but most contain concentrations higher than 80%. Use the data from the table to explain why. So we need to explain reasons why. So think about what hand sanitizer gels do. They kill bacteria. Okay, so effective hand sanitizers are going to kill bacteria and they will be working the best. So we need to link our information we've got from the table about the 60% and 80% ethanol and why that might mean that an 80% ethanol hand sanitizer is better than a 60% ethanol hand sanitizer. And obviously we have to understand and show that we need to link that to killing bacteria. And this question is all about membrane disruption. So we know that the ethanol is disrupting membranes. So disruption of those bacteria membranes is going to cause them to burst or die, absorb nutrients, whatever it is that's going to cause them to break those cells and damage them. So if we look at the table, we've obviously got the low absorbance at 60, but the much, much higher absorbance at 80 and 100. So the absorbance we have linked, and we've just explained in that question beforehand, that greater absorbance is because of more membrane disruption and therefore more substances leaking out of the cells. So high membrane disruption is going to be more effective at killing bacteria because it's going to disrupt the membranes more and therefore increase the chance of those bacteria dying because all their insides are going to come out through the membrane. So below 60% ethanol, there's very little disruption to the membrane as only a small amount of pigment was released. So that's us using information from the table. At 80%, there was a lot of pigment released, showing it would be more effective. Hand sanitizers with less than 60% would not disrupt the membrane of bacteria enough to kill them. And you could say might, you could say would, or you could flip that around, as you can see in the mark scheme, and you could explain and start talking about the fact that why 80% would be better. So you could link that to that idea that if you want an effective hand sanitizer that kills a lot of bacteria, if you use a higher concentration of ethanol, more damage to the membrane will be done. Environmental scientists completed an investigation measuring the biodiversity on 2 meadows. One is used for grazing local farm animals and one is left wild. Describe a practical technique they could have used to calculate the mean number of each plant species in each meadow. So this is a sampling techniques question. So obviously it's plants, so we're going to sample with quadrats and we're going to do it so randomly, and obviously don't forget to talk about using a large number of quadrats. And as always, we need to say that we would repeat it and calculate a mean. This is always a go-to mark and mark point in any practical exam question. This one especially though, because obviously the more quadrats you do, the more representative of the sample you're going to get of the population. So we need 3 marks. We're going to have to talk about being random, and you can say sampling randomly is enough to get the mark here, but often I like to explain or describe how I would actually do that, especially if it was a longer answer question. I would put a bit more detail into how I'm going to do the random sampling. So using a random number generator to produce coordinates, and then grid the meadow with tape measures to decide where to put the actual quadrats at random. Place a large number of quadrats in each meadow and count the number of each species in each quadrat, and then calculate a mean for each species in each meadow. Calculate the Simpson's Biodiversity Index, or D, for more 1, which is the no grazing, because obviously it hasn't been calculated. As we can see, there's a gap in the table. We've got the equation that's been given to us, which is handy. It's also reminded us of what little n and big N are. And it wants to give our answer to 2 decimal places. And that is because obviously the other D value in the table is also to 2 decimal places. So big N is the total number of organisms of all species, and we've got that calculated for us in the table, which is nice. Often you have to add that up and work it out yourself. So we can do our first row, which is capital N times capital N minus 1. So 93 times 92 gives us 8,556. And now we need to calculate our sum of little n times n minus 1. So for each row of the table, and often if there's space in the table, I literally do it like this so that I make sure I'm doing the right thing in my calculator, and you don't have to write it all out then. But 25 times 24 plus 32 times 31 plus 17 times 16 plus 2 times 1 plus 9 times 8 plus 8 times 7, obviously ignore the 93 because that was our big N. And if we do all of those and add them all together, we get 1,994. So now we can just do our division. So capital N value goes on the top, so 8,556 divided by our little n calculation, which is 1994. And then that's going to give us 4.29087 and some other numbers. And remember, we just want 2 decimal places, so we're just going to go 4.29. State the null hypothesis the scientists would use for the statistical test to compare the index of diversity in each meadow. So remember, a null hypothesis is a negative hypothesis, so it's always saying that there's no difference or there's no relationship or there's no correlation. And that is what a null hypothesis is. So it's kind of the opposite of what you think or expect to see when you do the test. So there will be no difference in the index of diversity between the meadows or in each meadow. The test returned a p-value of 0.032. Explain what conclusion the scientists made from this value. So it is less than our golden value of 0.05, so we're going to reject the null hypothesis as there is a significant difference here. But you need to make sure we explain that with the correct wording. So we can't just say it's significant and be done with it. We have to say it's less than 0.05, therefore it's a less than 5% probability that these results are due to chance. And that's what's going to get us that mark for explaining what that p-value means, because it's asking what conclusion we're making. So that's how we interpret the p-value. And then what the conclusion from that then is that there is a significant difference between the diversity index of each meadow. And what we can talk about is actually what that difference is. So there's significantly fewer or lower biodiversity, so fewer different individuals in the grazing field than the non-grazing field. And so it's always good to kind of not just say there is a difference, but actually say what that difference is, especially if you've got the data in front of you to support it. P-value is less than 0.05, so there is less than 5% probability that the difference is due to chance, so there is a significant decrease in the biodiversity in the grazed meadow. Explain why grazing had also reduced the biodiversity of animal species found in the meadow. So this is again about an explain question, so we need reasons why, but this time it's asking to give the reasons why biodiversity of animals would have decreased. So the table, remember, shows any plants, that's why we used quadrats to do the sampling. So this is like saying if the biodiversity of plants decreased due to grazing, why might the biodiversity of animal species also have decreased due to grazing? So this is quite a general answer that you can give to this question. It's always the same mark points. Factors that reduce biodiversity are reduced food sources, so reduction in food sources. You can't just say food because it's not about the amount of food. It's about different numbers of different species that can be eaten as food. So food sources. And obviously grazing, if animals are eating plants and grazing, that's going to remove plants or prevent plants from growing. So that's obviously a reduction in the food sources. And also reduced habitats. So there's reduced plant species, which means there's different or fewer places for them to live and hide in. And so we've reduced the number of food sources or the food sources and the habitats. And therefore, this is because we've reduced the plant biodiversity, and it's sort of that link between animals relying on plants as well. But any question that's asking about why biodiversity in general decreases in an area, there's always a mark for a reduction in the number of food sources and always a mark for a reduction in the habitats. And then that reduction in those causes animals to either go extinct or the population numbers to decrease or causes them to literally move away or leave the area. And so that is why the number of animals then decreases. That second point about the moving away or decreasing the population isn't needed for this mark here. It's quite a simple one. But if you were asked to explain why reducing food sources or habitats causes reduced biodiversity, then that is the reason why. The image below shows a cross-section of 2 blood vessels taken under a light microscope. State what type of vessel is represented by each letter and explain your answer using evidence from the diagram. So when it says type of vessel, it means is it an artery, a vein, or a capillary? And it's asking us to give a reason for our choice, but using evidence from the diagram. So not just a reason why we think it looks like that and just giving ideas about what the structure of the two vessels looks like. We have to be able to show it's something that we can see from the diagram. So for A, I can see that it's got a wider lumen than B. For B, I can see that it's got a thicker muscle wall than A. I can also see the folded, kind of wrinkled endothelium on the inside of B. And then obviously you can do the opposite. So narrower lumen for B and then thinner walls for A. So notice I've used like comparative, so thicker, thinner, because that's what we're going to have to use to, we're basically comparing the two to decide which one is which. So A must be a vein because it has a wider lumen. Okay, so just be sort of clear about that comparative element to your answer, because, or you can say the widest lumen of the two. And obviously we are kind of assuming neither of these are capillaries because we can't see kind of a one cell thick wall here. B therefore must be an artery because it has a thicker muscle wall, or you could have said it has the narrower lumen. But you've got options in the mark scheme you can see there, but those are the ones I chose. But as long as you've got A is a vein and B is an artery, and you've got a matching reason that makes sense, that is something you can see from the diagram. For example, you couldn't put A is a vein because it has valves, because you can't see a valve here. So you have to make sure it's something you can see from the diagram and that it matches the correct blood vessel and that you've got the blood vessels the right way around, and then you get the 2 marks. Describe and explain how an artery is adapted to carry out its function. So we need to say what structures does it have, what physical structural features does it have, and how does that specifically help it to carry out its function. So the main function of arteries is they're transporting blood at high pressure. So they need to both withstand the high blood pressure, so there's no damage to the artery, and they also need to maintain that high blood pressure to make sure it gets all the way to the parts of the body it needs to. So to withstand the high blood pressure, so to make sure they can cope with it, they have the thick muscle walls, and to maintain the high blood pressure, they have the narrow lumen. There's obviously lots of other sort of versions of this in the mark scheme, and they can sometimes be similarly linked to either withstanding high pressure or maintaining high blood pressure. But as long as you've picked one of the structural features and linked it to the correct function, then you can still get the marks. So the arteries have a narrow lumen to help maintain high blood pressure. They also have a thick muscle wall to help withstand the high blood pressure. So this is the exact language we've got to use, and this will be for any artery structure and function question that comes up. The answers and the language around the structure it has and the link to how it helps it carry its function is always the same. Describe the function of the coronary arteries. So it's asking us the function, so what do they do? I've put this in here without going through all the text first, although it does mention the coronary arteries in the text. We don't need that text to be able to answer this question. This is just a straight up, do you know this piece of content? So we'll do this first and then we'll analyse the text before the next question, because that is actually where it's needed. So the function of the coronary arteries, what do? They're going to transport oxygen, and specifically the coronary arteries transport that to the heart muscle or the heart cells. So they transport oxygenated blood, or they transport oxygen, or you can talk about glucose. All of that will get you that first mark. And then to the heart muscle, you have to have the word heart, and you can talk about muscle or tissue or cells, but it's the idea that it's going to the actual heart muscle that gets you the mark for mark 2. Foam cells are a type of phagocyte that contains high levels of cholesterol. Build-up of these can form a plaque that can lead to a narrowing of the artery, or atherosclerosis. So let's have a look at this paragraph quickly. It's basically saying that atherosclerosis, which we know is narrowed arteries caused by fat buildup, and we can link that to our knowledge of coronary heart disease. So we should hopefully have heard of this before. Apolipoprotein A1, or ApoA1, is a membrane protein that helps to remove fat molecules from within cells, including foam cells stuck in the walls of arteries. Dysfunction of this membrane transporter is associated with cellular cholesterol overload. So when this membrane transport protein, ApoA1, doesn't work, then there's too much cholesterol builds up in cells. So cells contain more fat. And this includes the foam cells, which it's said have high levels of cholesterol, and a buildup of those cells can form that plaque which causes atherosclerosis. A controlled study looked at the effects of smoking and coronary artery disease on the presence of ApoA1 in blood plasma. Their results are shown in the table. So potentially we're expecting to see the fact that people with coronary artery disease might already have atherosclerosis. They might already have that narrowing of the arteries. So we've got data. So we've got non-smokers, and obviously before quitting smoking, there isn't they aren't smoking, so they're not going to have that need to quit smoking. But they've got quite high, 1.7 grams per litre of ApoA1. Then we've got smokers that don't currently have coronary artery disease. Their ApoA1 is already lowered, it's 1.33. And then we have smokers with coronary heart disease, which is higher, so that's 1.38, but still lower than people who don't smoke. Then we've got them not smoking for 90 days, and we can see for in both cases with the smokers, it increases their ApoA1 by quitting smoking for 90 days. And then when they restart smoking after 90 days again, so another 90 days, so 90 days of not smoking, and then they restart, and then they smoke for 90 days, then we've got this decrease again. You see the ApoA1 decreases. It doesn't quite go back to what it was before they stopped smoking, but it obviously decreases. So we're saying that more of ApoA1 means less fat in cells, specifically the foam cells, which can cause the buildup and cause the atherosclerosis. And it definitely shows here that we've got an increase in ApoA1 when we do quit smoking. So the first question is just about suggesting one factor that should have been kept consistent across the groups. So this is a clinical trial-based question, and this often comes up where they will ask you what's something they should be kept the same about the groups in terms of the people in the group. So what factors could have contributed to maybe different results that they should have tried to keep the same. You're not going to be able to keep everything exactly the same, but have a think about what might be different between different people that might cause the results to not be consistent. The main general answers, which are ones you can pretty much always guarantee and give for these if they haven't mentioned them already, is sex, age, and then health conditions. So the sort of differences between men and women, differences between different age groups, and then potentially health conditions. So if they're already sick or they've got other health conditions that could interact, that should all be considered. Here we've also got the element of smoking. It says both of those groups are smokers, and then it's with and without coronary heart disease, but it doesn't say how long they have smoked for or how many they smoke a day. So something like that should also be considered, because obviously the frequency of the smoking and the historical length of time that they have been smoking for could also contribute to how much atherosclerosis they could potentially already have, whether they have coronary heart disease or not. So something like that where you can see something that's within the data that you should obviously keep the same. I'm going to go for sex and age of the participants because that's a nice, easy general one. You've obviously got the options in the mark scheme there that you can use. So part E says, evaluate how well this data supports that stopping smoking can reduce the risk of atherosclerosis. So with an evaluate question, you have to say the pros and the cons, or the fors and againsts. And we've got to get that from data in the table. So what from the table shows you that whether the data actually supports this or not. And it's asking about stopping smoking. So we need to look at the impact of stopping smoking, and can that help to reduce the risk of atherosclerosis, which is the fat build up in foam cells that could potentially then cause them to build up and create that narrowing of the arteries. So if we reduce the fat in the foam cells, there's potentially a reduced chance that they could build up and cause that blockage. The main thing here is obviously we know that we're looking for smoking shows that there is, if you stop smoking, then there's an increase in ApoA1. It shows that people who smoke have lower ApoA1 than the people who don't smoke. And then we've got to sort of think about, what does the data show in terms of, do we think that that's enough? And do we think that it definitely supports the fact that if you stop smoking, it's going to reduce your risk? What does the data tell us? So we have got data from people who already have coronary heart disease. So they potentially already have atherosclerosis. So their risk of developing it, or they might already have it, we don't know. But there's a chance, that obviously a greater chance that they are going to have that already or be at more risk of it. The main things with any data question like this, when you're asked to say, does it support it? Does it prove a conclusion? How far does it support this idea? Is look at the sample size and think about statistics. So we've got a very small sample size here, literally 17, 30, 28. 2 out of 3 of those groups are under 30, which we would say is a very small sample size, especially for humans and data on humans. And there's no stats test, there's no standard deviation, so we don't know if any of these changes we're seeing in the ApoA1 levels are due to be significant or not, because we don't have any of that data. There's also no direct link seen here in the data between this idea of an increase in ApoA1 and actual decrease in atherosclerosis. They didn't measure atherosclerosis or its development. So we are just going based on the fact that we've got this idea that obviously the protein that we're talking about, if we increase it, that should help reduce fat in cells, but we don't know that that's directly going to cause the change that we want to see. So I've written my answer here. There's lots of marks, as you can see in the mark scheme, and we just have to make sure that we talk about both the supporting and the disproving, and most of those kind of negative marks can come from talking about the small sample size, no standard deviation or statistical test, and also just kind of our understanding of the link between the information we've got given here in the first part of the question and the data in the table. So the data does show that stopping smoking resulted in an increase in ApoA1 concentration in the blood in people with and without CHD, or coronary artery disease, that should be CAD. An increase in ApoA1 would help to remove more lipids from foam cells, which may help reduce plaque buildup in the arteries. So there's me saying, yes, it shows an increase in ApoA1 after stopping smoking, and there is a potential link between the increase in that protein and how it might help reduce plaque buildup. But there is no direct measure of a link between increased ApoA1 and atherosclerosis reduction. The greatest increase was seen in people who are already suffering from CHD or CAD, and so they may already have atherosclerosis. So this is me saying, like, 2 things that support it, so a thing that supports it and then explain why that could reduce the risk, but we've got no measure of a direct link to prove that, like causation, and we don't know that obviously the biggest increase we saw was in the people who already have coronary artery disease, so there's a potential there that we don't know if they've already got atherosclerosis or not, and if it could reverse it. In smokers without CHD, the increase in ApoA1 was less than in people with CHD when they stopped smoking for 90 days. And in both cases, the increased ApoA1 was still lower than in the non-smokers. So the risk will not be reduced entirely to the same as if they had not smoked. So although we're saying there's a potential there that it does increase the ApoA1, and smoking would normally decrease that, which we can see from the data, stopping smoking doesn't entirely bring their ApoA1 back up to the levels it would have been if they'd never smoked. But again, we don't know, because this was only for 90 days. So we do not know if that change would have continued if they'd stopped smoking long term. We don't have that data, and you can say that. This data was collected from a small sample size, and there is no standard deviation or statistical test to show if the differences in ApoA1 are significant. There we go. And that's always something, remember, that you can say and point out if you've not got any statistical tests or standard deviation on your data. And then obviously if you do, you can talk about whether it shows significance or not. But in this case, we didn't have any. So we can say that as a way of saying we don't know how significant these results are. The graph shows the change in oxygen concentration in the water and blood as they flow along gill filaments. So this is a gas exchange in fish question. So we've got the water, and you can see that as we move along the gill, the concentration of oxygen in the water decreases, and as we move along the gill, the concentration of oxygen in the water increases. Use the graph to explain how this system leads to efficient gas exchange in fish. So hopefully we know this is talking about the countercurrent flow system. What does the graph show about the concentration gradient? What can we get from this graph? And it's about gas exchange, so it's about the oxygen diffusion. And the efficiency, you can see here, it's about talking about the fact that this system maximizes diffusion of oxygen across the whole length of the gill. The countercurrent flow system means that water and blood flow in opposite directions. This is a sentence that we always should be writing when we're answering these questions. Okay, that's the first sentence we should always be writing. So you can see here, obviously, as the decrease in oxygen of the water happens, the increase in oxygen of the blood happens. So oxygen is always moving from the water into the blood along the full length of the gill. So it doesn't reach equilibrium. The concentration of the oxygen in the water keeps increasing, in the blood, keeps increasing. And so that means that we have maintained the concentration gradient, because if you didn't maintain the concentration gradient, diffusion wouldn't keep happening across the full length. As the concentration of oxygen in the water decreased, you wouldn't get that. You'd get equilibrium eventually. So the countercurrent flow system, the opposite directions, means that the concentration gradient of oxygen is maintained. So oxygen always diffuses from the water into the blood along the full length of the gill. And this is a standard answer to this question. If you just had the question, explain the countercurrent flow system in PhET, or explain how the countercurrent flow system makes efficient gas exchange in gills, this would be your answer. It's the same 4 marks and the same answer, regardless of whether there's a graph or not. This is what we have to say. We have to talk about the countercurrent flow system. We have to say what that means about the opposite directions, the fact that that maintains the concentration gradient, and what that means. It means that there's always a higher concentration of oxygen in water flowing next to a lower concentration of oxygen in the blood. So it always moves from the water to the blood, and that happens for the whole length of the gill filament. And that means that you've got maximised diffusion. Fish have mucus-secreting goblet cells on the tips of lamellae. The mucus contains a high concentration of glycoproteins. Describe how glycoproteins would be produced in the cell. Do not include details of protein synthesis in your answer. So we need to explain how, we don't need to explain how the polypeptide was made, we just need to explain how the polypeptide gets processed into a glycoprotein. So make sure you always listen when they give you instructions like this, because it's only a 3 marker and we'd expect a longer answer question just for protein synthesis. So it's clearly saying it doesn't want you to focus on that. It wants you to focus on how inside the cell we get from a polypeptide to a glycoprotein. So a glycoprotein is a protein that has had a carbohydrate chain attached. This is something we should know. So we're just talking about how and where in the cell that happens. So the protein is going to be released, or the polypeptide will be released from the ribosome. It's going to go to the Golgi, because that is where proteins are modified. And that is where we will attach a carbohydrate chain to the protein, and that becomes then a glycoprotein. The polypeptide chain leaves the ribosome in the cytoplasm and is transported to the Golgi body. In the Golgi, a carbohydrate chain is attached to the polypeptide, forming a glycoprotein. So you've got alternatives in the mark scheme, like talking about it leaving the rough endoplasmic reticulum instead of the cytoplasm. That's where the ribosome is. Just thinking about cell structures, where in the cell does this happen, where does it move to, and what the cell functions are for the parts of the cell that are involved in this process. Flavobacterium is a genus of filamentous bacteria that causes bacterial gill disease in fish. Bacterial gill disease causes excess mucus to surround the gill filaments and can cause the lamellae to fuse together. Suggest why more mucus is produced when fish are infected with the bacteria. So this is a suggest question. So we're going to use a bit of our own knowledge. So what do you know about mucus and pathogens, think about it. Why do we produce more mucus when we have a cold or potentially being infected with pathogens? Things like hay fever, where you get a runny nose and things like that. So what links this idea of mucus and pathogens? What do we know mucus does to pathogens? It traps them. So that's why we have we have goblet cells producing mucus in all of our airways. So we have mucus to trap them, and then normally we try and get rid of that mucus. But obviously fish can't kind of cough that out and stuff like that in the same way we can. But if you've trapped the pathogens, that means they're more likely, or it's easier for the phagocytes to come and destroy the bacteria. So that's where I would go with this answer. It's quite a hard one for 1 mark, but you could also think about the fact that the mucus forms a protective coat or a protective layer that might help to protect the gills as a defense against toxins or something the bacteria might cause damage. That is also marked in there as well. So with these suggest questions, there isn't always one specific correct answer because it's down to a little bit of interpretation and what you've used your knowledge to come up with. Explain why fish with the disease face into the water current for increased water flow over the gills and exhibit exaggerated opercular movements. So basically, why do the fish change their behaviour to increase the water flow over the gills? So it tells you that in the question. So we are just explaining why that happens when they have the disease. So this is the equivalent for thinking about the exaggerated opercular movements. This is the equivalent of us kind of being breathless when we are ill with it or have some kind of lung issue. So why might you need to increase your breathing rate or try and in this case increase the water flow? Because you're trying to get more oxygen because gas exchange has been reduced in some way. So why do fish with the disease have a reduced gas exchange? So if you think about back to the start of the question, there's always clues in the question to help. It's where it says the lamellae can fuse together. Okay, so if they fuse together, instead of being lots of fine, thin, branching structures, they fuse together in big clumps, that's going to reduce their surface area and therefore reduce oxygen diffusion. Okay, you've got alternatives in the mark scheme to think about the mucus or this idea that if they fuse together, they're thicker, so the diffusion distance is longer. But it's got to be something that's going to affect the rate of diffusion of oxygen and therefore reduce the diffusion of oxygen that's coming in to the blood. And that's where they're trying to increase the amount of water flow over the gills to try and maximise that oxygen delivery. So I've drawn a little sketch of my kind of lamellae, my filamentous branches, and then what happens if they all stick together and form like clumps. We've reduced our surface area, just so you can kind of visualise what we're trying to talk about here. So this was about finding clues in the question, understanding why they might need to increase the water flow, and then being able to link it to the disease and link it to oxygen diffusion. So the fusing of the lamellae has reduced the surface area, so there is less oxygen diffusing into the blood. Many organisms display adaptations to their environment, and sometimes organisms that depend on each other have evolved adaptations together over time. One example is the peccary and cactuses in desert habitats. So just in case you're not sure, the peccary is the pig and there is the cactus. The table below shows some different adaptations of both organisms. Tick the box to show the correct type for each adaptation. So the cactus being covered in spines, that's part of the plant, so it is part of the plant's anatomy. Peccaries eating cactuses with fewer spines, that's a behavioural choice. Cactuses producing oxalic acid, which can cause diarrhoea. That's a chemical, so it's something that happens within cells that is produced, so it's a part of its physiology. And peccaries having a specialised structured digestive system is going to be, again, part of the anatomy of the peccary, part of the body, so that is anatomical. So we've got anatomical for the digestive system and the spines, we've got behavioural for the choice of eating the cactuses, and then we've got the physiological for the oxalic acid. Apart from spines, explain 2 other adaptations of the cacti to the desert habitat. So this is an adaptations of xerophytes question. This is thinking back to like adaptations that plants have to the desert conditions to reduce water loss. But we need to make sure that we don't just state what the 2 adaptations are, we explain how it helps them to survive. So I'm going to go for thick cuticle that is waterproof and fewer stomata, so less water is lost through transpiration. You can pick obviously any two from the long list that's in the mark scheme, just as long as you make sure you've matched it to the correct explanation of how it's an adaptation. As well as having large salivary glands, the peccaries have a specialised forestomach that contains bacteria that digest the high cellulose content of the cactus. In some species, these bacteria have adapted to also be able to degrade the oxalic acid. Explain how these bacteria could have developed. So it's saying that the bacteria have changed over time to be able to do something new, which is to degrade the oxalic acid. So this is just a very fancy, dressed up natural selection question, because if it's the bacteria have adapted and therefore changed over time to a selection pressure, which is going to be the oxalic acid, which obviously would be acid, it would be pH, it would be something that they're not used to, they are then able to have overcome that through the process of natural selection. So there'd be variation caused by mutation, which means that some of the bacteria could cope with the acid and they're going to be able to survive and reproduce, and they will pass on these genes that enable them to do that to their offspring, and eventually we'll have a population of bacteria in some of the pigs that will be able to do this. So some stomach bacteria have a mutation that enables them to break down the oxalic acid. These bacteria are more likely to survive the low pH and reproduce. I've added the low pH in there. You didn't necessarily have to have that in there, but the idea that they can survive or they can somehow overcome the selection pressure and therefore they're able to not be killed by it or not be outcompeted by other bacteria or whatever it is. And therefore they're going to pass on their genes to their offspring. And for 3 marks, that's all the detail for the natural selection we need to go into, because it's just explaining how we have some populations of bacteria that can able to do this now. As a response to increasing herbivory by peccaries, cactus spine number has increased. However, there are also a parasitic insect that is more likely to lay its eggs in the spines of cacti that have more densely packed spines. So we've got kind of 2 different things going on here. If you have more spines, then you're less likely to be eaten by the peccaries if you're a cactus. But if you have more spines to try and prevent that, then you're more likely to have this parasitic insect lay eggs inside the tissue of the plant. So you don't kind of want either of those to happen. The graphs show how the frequency of cactuses with different spine numbers in the population has changed over time. Graph A shows an earlier survey and graph B represents a more recent survey. So we've got before and then now, or more recently, we've got the spine number on the x-axis, and therefore we've got the number of cacti with that spine number, the frequency, on the y-axis. Use the graphs to describe the type of selection that has taken place in the cacti population. What can you see? So what are we going to be able to see in the graphs that is going to help us to describe the type of selection? So that's when we talk about describe questions, we say what we see. So is it directional or is it stabilising selection? Okay, so we need to describe the type of selection that we can see from this graph, and we need to give evidence from the graph basically to back up that point. We're not explaining how the selection has happened or anything like that for this point. We're just saying what can we see in the graph, evidence from the graph that will help us choose which type of selection and say why it's that type. So the range has decreased. So it was about 55 to 155 ish in graph A, and that's reduced now to between about 70 and 130. So that range has massively decreased, and we can see that. The mean has stayed around the same, similar numbers, so about 100, just below 100, and just below 100 basically for both. But you'll notice the y-axis is slightly different. So the frequency of around 100 spines, the number of cactuses was about, well, it was less than 100 or just above 100. Whereas for the more recent survey B, we've actually got an increased frequency. So there's more cacti with that mean number in the new graph. So we've got a similar mean, but the frequency of individuals with that mean spine number has increased. So hopefully this should lead us to the conclusion that it's stabilising selection, because we've got few at the extreme survived, and that's why our range is decreased, and clearly more are surviving around the mean number, which is why that frequency increased. So that all of that information is enough from the graphs to get us to 3 marks here. The graph shows that stabilising selection has occurred because the range of number of spines has reduced. So I'm going to get a mark for stabilising. I'm not going to get a mark for the range though here because I didn't include data from the graph. So if you're going to talk about the range decreasing, you should talk about the difference in the data. I'm going to get a mark for saying the mean has stayed very similar. Okay, so that will get me a mark around 100. And the frequency of cactuses with the mean number of spines has increased. Okay, so I'm going to get a mark for stabilising. I'm going to get a mark for talking about the fact that the mean stayed very similar, around 100. I did quote that number, so I get that mark there. And the frequency of cactuses with the mean number of spines has increased, so I'm going to get that mark. If I talked about the range and actually given some data points, numbers, then I could also have got that range mark. Because it says you use the graphs, you should always try and quote data. And that was my fault for not doing that. Explain why these changes in the cacti population have occurred. So now we're explaining why we saw all those changes that have showed us that stabilising selection is happening. So why was it happening? So we go back to the start of the question and that information it gave us about the 2 different selection pressures. So the idea of the peccaries and then also the parasitic wasp. These 2 are 2 selection pressures that cause the mean to be more favourable, and you're more likely to survive if you're somewhere in the middle than if you have too few spines or 2 more spines. So the mean clearly increases survival, mean number of spines, because that has the greatest frequency, and all the ones that had the extreme, so too few spines were eaten by the peccaries, and too many spines had too dense spines, and therefore they were parasitised by the insect. So that is basically all we need to say for 3 marks. So we need to talk about the selection pressures, we need to say the difference between the 2, and then say that having the mean number of spines was more favourable and more survived, and therefore passed on those alleles to their offspring. So the peccaries and the parasite are both selection pressures. Too few spines means cacti are more likely to be eaten, and too many spines means they would be parasitised. This reduces survival. More individuals with a mean number of spines survive to reproduce and pass on genes to offspring. There we go. 3 marks. Now this is my kind of favourite part of paper 1. This is what I call the gift questions, which are lovely because they are long answers, but they tend to be quite simple, describe, explain, or compare questions where they're just asking you to tell a process or tell content that you know in detail for a decent number of marks. And if I was you, and this is the recommendation I give to everyone in the exam, when you get into AQA paper 1, you sit down and you go straight to the back and you look at these 2 questions first and potentially do them first, or at least brain dump a little plan. You'll see how I'm going to sort of sketch my plan out, or at least put some information down that I can come back to. But they're easy marks at the end of the paper, so don't run out of time and potentially waste sort of 10, 11 marks on this bit of the paper. Definitely come back, do this one first, and then you can always come back and check it at the end, but you don't want to run out of time and have to rush these, because these could be a lot easier than some of the application questions you might find earlier on in the paper. Compare the structure of triglycerides and phospholipids. So whenever we're comparing, we have to give the similarities and the differences, and it wants the structure to be compared. So the bonds, the molecular structure of these 2 molecules. So a quick sketch of a triglyceride. It's got 3 fatty acid chains, 1 glycerol molecule, and 3 ester bonds. Whenever it asks about structure of a molecule, bonds are always going to be important for marks. Then I'm going to draw a phospholipid. So I've got 1 phosphate group, I've got a glycerol molecule, and then I've got 2 fatty acid chains, which means I've only got 2 ester bonds. So now we can see the similarities and the differences, and we've just recapped our knowledge. It's about writing comparative statements. So they both have this and they both have this, but this one has this and the other one does not. Making sure you're clear which structures have what parts and the similarities between them. Both molecules contain fatty acid chains, a glycerol molecule, and ester bonds. They're my 3 similarities. You could have talked about the fatty acid chains as well, but these are the main 3. In phospholipids, there is a phosphate group bound to the glycerol molecule, but not in triglycerides. So I've made clear to say this one has this, but it's not present in the other molecule. In triglycerides, there are 3 fatty acid chains, but only 2 fatty acid chains in phospholipids. Again, this one has this, but the other one is different in this way. Explain the steps in meiosis that lead to an increase in genetic variation. So this isn't explained, so we do need reasons why in this one, but it is talking about the steps in meiosis, so what happens in meiosis. And then how those stages of meiosis increase genetic variation. So I've got to mention homologous chromosomes because that's going to be a key bit of language or a key word here. And obviously the homologous chromosomes do 2 things to increase genetic variation. They can do crossing over, which happens in prophase 1, and that creates new combinations of alleles. And there's independent assortment in metaphase 1, which is where we're going to randomly split which maternal or paternal chromosome is going to go into each gamete by how they line up in the middle. So that's basically what I'm going to do for 5 marks. I'm going to talk about each of these 2 processes, when it happens, what happens in that stage, and how it creates genetic variation. So in metaphase 1, homologous pairs of chromosomes are independently segregated, or you could say assorted. So I've said when, and I've said what, and now I have to say how does that increase genetic variation. It randomizes which chromosome, maternal or paternal, will be present in each gamete. In prophase 1, crossing over between chromatids of homologous pairs leads to alleles being exchanged. This creates new combinations of alleles on each chromosome. Okay, so for each of these 2 stages of meiosis, I've talked about what the stage is, what happens, and then I've made it clear how that increases genetic variation, either by randomly assigning the maternal or paternal chromosomes and therefore their alleles to each gamete, or creating new combinations of alleles on each chromosome that will then end up in the gametes.
Ouch! This is why in some videos I explain scratches.